Good morning, good afternoon. Thank you for the opportunity to present today. So my name's Adam Winterhalter. I'm the Business Development Manager for Sysmex Australia for flow cytometry. So today I'm going to talk about DNA ploidy and its application in the aquaculture industry. So firstly, what is ploidy? So the definition of ploidy is the number of sets of chromosomes in a cell or in the cells of an organism. And this is usually denoted by the letter N. So N refers to the number of sets of chromosomes. So 1N equals a haploid organism. 2N would be a diploid organism. 3N, triploid, 4N, tetraploid, and so on, up to multiple levels of polyploidy. On the right, there's an image showing a haploid, a diploid, and a triploid organism. So the haploid has one set of chromosomes, the diploid has two sets of each pair of chromosomes, and then the triploid has three sets of individual chromosomes. So mitotic division results in the chromosome duplication within a cell, and this results in the even division of DNA between cells to maintain the level of ploidy. So the mitosis is shown on the right-hand side there in red, where we have a 2N organism divide and replicate its DNA and then divide into two cells so that then we have duplicate copies with even numbers of chromosome in both copies. Meiosis results in a mitotic division of the cells, and that meiotic division halves the genetic material, and this happens in spermatogenesis or oogenesis, where we're creating sperm or eggs for fertilization and creation of a new organism. So how do we measure ploidy? So ploidy is commonly measured by two methods. The first is chromosome counting by microscopy, which, as you could imagine, it's very time consuming, it's labor intensive, and it's very subjective and quite challenging to see the chromosomes and count the number of chromosome pairs that are present. The other method that we'll focus on today is flow cytometry. So flow cytometry, it's fast, it's very easy, and it's reproducible. And this is an example of what a flow cytometry plot looks like, where we have a peak that designates the ploidy level of the organism that's being analyzed, and then a smaller peak that shows the dividing mitotic cells. So this might be a diploid, and then the mitotic cells show twice as much DNA content or fluorescent staining as a surrogate marker for the DNA levels. So for flow cytometry analysis, what does it require? Well, firstly, we need to extract the nuclei from the cellular tissue. Once we've extracted the nuclei, we then need to stain the DNA within the nuclei with a fluorescent dye. And this needs to be a stoichiometric dye that saturates the DNA. So it's really important to saturate the DNA, otherwise we won't have consistent staining when we run the samples by flow cytometry. Once we run the samples, we then measure the level of fluorescence on a flow cytometer, and then we compare that level of fluorescence to known control materials that have known levels of ploidy. So we need to run a diploid control, a triploid or a tetraploid control, or a haploid control if that's what you're looking for, so that you can set regions and gates on the flow cytometry plot are representative of the DNA level within the organisms that you're sampling or running. In terms of a sample, you can run any sample on the instrument providing you can extract the nuclei from that. So it might be a piece of leaf tissue. If you're running plants, it could be a piece of root tissue, or from a fish or a shellfish, you could use some of the gill tissue, which you can then macerate, extract the nuclei. So you want to chop them up to release the nuclei, and then stain it with dye and run it on the instrument. So the piece of tissue, as a rough guide, should be about 0.5 of a centimetre squared in size, and that should be adequate to extract sufficient nuclei for analysing by flow cytometry. So Sysmex has a range of products that meet the needs of the customer for flow cytometry. So we have the hardware, the Ploidy analyzer. We have reagents, a variety of cystine reagent kits, and we have consumables such as Celtrix filters and DNA control material. So the Ploidy analyzer is available as three different models. So we have laser choices as a UV only light source, a 532 nanometer laser only light source, or a combination of both the UV and the 532 nanometer laser. There's also an optional autoloader for the SciFlow Ploidy analyzer should you want to do high throughput screening of samples. So the SciStain reagent kits, we have a variety of reagent kits based on the different light sources in the instruments. So we've got a UV Ploidy kit, which is mainly used by the fishery and oyster customers. We've got UV Precise P and a UV Precise T kit. So these are a two-step kit for first extracting the nuclei and then a second step for staining the nuclei. And then the same kits with PI as the dye, where we have PI absolute P and PI absolute T. And this is more for looking at genomic size, so the absolute size of the genome in both plants and tissues. And then finally, the consumables, we have Celtrix filters. So the Celtrix filters are extremely important in Ploidy analysis. So once you stain and process the sample, you need to filter it through the filter to remove any cellular debris from the sample prior to running on the instrument. Not removing debris will result in the instrument being blocked and failing to run sample. We also have DNA control, which acts as a QC material, and you can run this daily to demonstrate the instrument is performing correctly before running your unknown samples that are being tested. So what dye should I use? So there's two different dyes that are available. We have DAPI, which is the 4,6-diamidino 2-phenylindole, and PI, which is prapidium iodide. So DAPI and PI, much easier to say. So DAPI is a UV excited dye, so it's excited in the 355 nanometer range. And looking at the spectral chart underneath the table, we can see that the blue dotted line shows the excitation profile of the DAPI fluorescence dye. So the peak excitation for DAPI is somewhere around that 355 to 365 nanometers. Upon excitation, DAPI fluoresces in the 450 to 470 wavelength range. So that's peak fluorescence for DAPI. So DAPI is unique that it only binds to the A and T residues in DNA. And because it only binds to A and T, it means it doesn't bind to RNA. So when we're processing samples, there's no need to add RNAs to digest the RNA in the sample. So this makes for much quicker sample preparation and much cleaner sample preparations. DAPI is very good for Ploidy analysis because it does give such a clean signal. However, you can't use it for genome sizing because it only binds to the A and T residues. So if you're comparing genomes between two different species, the A to T ratio changes between species. And this would mean that the level of fluorescence that you see of the bound DAPI dye would also change. So you can't use DAPI for genome sizing. What you can use for genome sizing, though, is prapidium iodide. So PI, when we look at the fluorescence profile below, its peak excitation is actually down around 300 nanometers, but commonly it is excited at 488, which is a blue laser, or around 532, which is a green laser. So we choose the 532 nanometer laser rather than the 488 because 488 also excites chlorophyll when looking at plant tissue. And this can lead to a high background fluorescence when you're trying to dissect the DNA signal from the background chlorophyll signal. So using the 532 nanometer laser is better because it does not excite chlorophyll. So PI, unlike DAPI, binds to the histones that the DNA and RNA are bound around. So it's a very good measure for genome sizing. It can be used for Ploidy analysis, although not as clean as DAPI because we have to add RNAs into the mix. And as I said, RNAs is required for sample preparation to digest any bound PI that's bound to the RNA so that we get a clean DNA peak when we're looking at either the genome or the Ploidy level of the organism of interest. The peak emission for PI is out around the 630 nanometers. Okay, so let's look at the aquaculture industry in Australia. So aquaculture is the fastest growing of the animal and food producing sectors in Australia. It's increasing at an average rate of 4.3% per year for the past decade. This is information circa 2019. So the aquaculture industry is valued at about 1.4 billion Australian dollars, and it's expected to grow by 7% in 2022. And these figures aren't unique to Australia. We see similar figures around the Asia Pacific region, where fish farming and seafood farming is starting to take off. So the common aquaculture species include oysters, Atlantic salmon, prawns, mussels, abalone, barramundi, yellow-tailed kingfish, and southern bluefin tuna. So they're the main species that are farmed in Australia. In Australia, the salmon industry is valued at about 497 million Australian dollars, and the oyster industry from farming is valued at about 106 million Australian dollars. So the farmed oysters in Australia are the Sydney rock oyster and the Pacific oysters, and they make up 90% of what's farmed, and then there's a few flat oysters and tropical oysters that are farmed as well. Now, the reason I list the salmon and oyster industry is because these are the only two that currently use Ploidy in their operations. And the Ploidy measurements are used for different reasons in these industries, which I'll discuss shortly. So where can Ploidy be used? So as I've mentioned, it's possible to use it in aquaculture when we're looking at oysters and Atlantic salmon. And a new emerging industry is farming of abalone, and they're currently investigating if Ploidy has similar effects on abalone as it does on oysters. Agriculture is Ploidy is heavily used in agriculture, so it can be used in flowering plants to confer specific traits. It can be used in crop plants and also fruits. So why is Ploidy important in these different applications? Well, different levels of Ploidy can confer different outcomes. So higher number of chromosomes can confer drought tolerance. You can get disease resistance by having a higher Ploidy level. You can have tolerance to the environment, where it might be a high salt environment or a high heat environment. There's survival advantage to organisms and plants. Ploidy can be used for sterility, so you may not want a plant to cross-pollinate native species of a similar type, or you may want to avoid getting seeds in your bananas, for instance. So bananas are all triploids and they're sterile because you don't get seeds in them. Different levels of Ploidy can also confer faster growth rates or increase protein yields, etc. So there's various reasons for applying Ploidy to different agricultural and aquacultural applications. So why is Ploidy important to oyster farming? So Ploidy is used in the oyster industry to improve yields and profitability, so it's purely economic. So Sydney rock oysters, when they're naturally occurring, are diploids, and they reach maturity in about 3 to 4 years. So that's an oyster that's about 40 to 60 grams in size. So it takes 3 to 4 years to reach maturity. The Sydney rock oyster, when it's farmed and genetically manipulated to be a triploid oyster, reaches maturity in half the time, so 1.5 to 2 years. So for the farmers, this means they can grow out and sell twice as many crops using the triploid oysters as they could if they stuck with just naturally occurring diploid oysters. So the economic benefit is quite huge to the farmers. In the salmon industry, Ploidy is used for a different reason, and this is for sterility. So the salmon are not native to Australia. Atlantic salmon are an introduced species. So they want to make sure that they can't breed with a native salmon or trout that exist in the same waterways where they're farming the Atlantic salmon. Triploid salmon also grow to maturity faster because they're not putting energy into producing the sperm and eggs, and it all goes into producing the meaty body of the salmon. So during the life cycle of an oyster, Ploidy is measured at multiple points. And this is an example of what the life cycle of an oyster looks like. So we have the adult oyster. that the farmers, or the breeders, I should say, not the farmers. So the breeders have mature broodstock oysters, which they screen for ploidy levels, and then they select them for breeding. So they want diploid males and tetraploid females. So this is males with 2 copies, females with 4 copies, so a 4N female. And then they mix them together so that they harvest the oyster sperm and the oyster eggs and mix them together for fertilization. So when they cross the 2N and the 4N, they end up with a 3N batch of fertilized eggs. So those fertilized eggs within 24 hours turn into free swimming larvae. And then at this point, they can actually measure ploidy to gauge the success of the breeding. So if they get the breeding right, they should have close to 100% of the larvae as triploids. Once they grow out, so in another 3 to 6 months, they then test the oyster spat, and this is as a quality measure before they sell it to the farmers. So the farmers want to know what percentage of the oyster spat that they're buying are triploid. So it's a quality control measure for the farmers. And then once the farmers buy them, they grow them out in their baskets to maturity, which is around 18 to 24 months, and then they sell them to the markets. So the farmers themselves, they don't test for ploidy. It's only the oyster hatcheries that test for ploidy. So I hope this presentation has given you a good understanding of how science and ploidy can be utilized to improve farming, to improve aquaculture applications, and to have an economic benefit for the organizations that are adopting the scientific techniques for ploidy. So if you have any questions now, please feel free to ask them. I'll be happy to answer. Thanks very much.